Hi everyone, my name's Andrea Henle, and I'm an associate professor of biology at Carthage College. I'm here today to tell you about some research that I've been working on with a student of mine, Melanie Goussois. Melanie's an undergrad who'll be graduating from Carthage this upcoming spring, and she also has a lightning talk here at ASGSR, so I encourage you to look out for that. So I'll be telling you about how we used a database called TOAST to engage undergraduate students in the mining of bioinformatics data from NASA's GeneLab repository. First, I want to introduce to you the course that I've been offering at Carthage College the past 2 years, and in which I was able to engage the undergraduate students in this bioinformatics research. So this course is called Space Biology, Human Health and Disease from Space to Earth. And there are several questions I try to answer in this course throughout the semester. One of those that we try to help the students understand is more of the history of space travel. So what have we learned about human biology from the time we've spent in space these past several decades? We also focus a lot on human biology and how that helps us understand diseases here on Earth. So how do these experiments that scientists are currently conducting, how do these help us understand disease here on Earth? So things like vision loss due to changes in fluid shift in the head, muscle atrophy due to microgravity, changes in cell shape, size, and growth of cells, changes in bone density, all of these types of conditions that we try to understand what's going on in space. They can help us understand similar diseases here on Earth. And lastly, we talk about how the ongoing research in space on the International Space Station will help us prepare for future human adaptation to space, and how some of that research is even being conducted here on Earth using simulation studies or ground-based studies. So I want to focus most of today's talk on the laboratory experiments that the students do, and tell you a little bit about how bioinformatics plays a role in this lab work. So one of the objectives for the course, particularly for the lab, is for students to be able to engineer a 3D printed payload of sorts that's going to allow them to do gravitropic and phototropic assays on Arabidopsis thaliana, the most commonly used model plant. This is a picture of the 3D device that they print. It's called a Flash Labs. And there's a very similar one for sale now by a company called COSI Incorporated, which has been founded by my collaborators at Madison. But essentially this 3D printed device allows them to have a small camera that's going to do time-lapse photography of these petri dishes here where the Arabidopsis seeds are growing. And around the outside of the dish, there's also an LED light source. You can change the color to have different colors of light and test how that affects the plant growth. And it also contains a motor so you can rotate the direction at which this plate is oriented, which will essentially introduce a change in gravitational vector. So you can see here, Arabidopsis seeds should normally grow with the roots down with the force of gravity, the shoots growing up towards the light source. You can rotate the plates 90 degrees in a gravitropic assay, and you should be able to see if the plants are working normally, like this wild type plant, that the roots turn 90 degrees with the direction of the new gravitational force always going down here. The students then can analyze the change in that root tip angle after they rotate the plates, and they can look at them in wild type plants, so it should be close to 90 degrees or perfectly 90 degrees. And then they can also look at plants that have mutations in certain genes that they think might be responsible for gravitropic sensing in the plants. And they can see how does that affect the change in the root tip angle. One of the main goals that I wanted my students to start to do is to use bioinformatics databases to identify candidate genes that might be involved in the microgravity response. One of the tools that they use is called TOAST. It stands for Test of Arabidopsis Space Transcriptome, and this is a tool that Richard Barker and Simon Gilroy at Madison developed. It essentially is a database that makes all the data from NASA GeneLab really user-friendly, particularly the data from plant experiments that have been deposited in GeneLab. So with TOAST, you can mine all of this data from GeneLab studies, and you can start to identify mutations that you might want to study further. So we could order an Arabidopsis seed with that particular mutant that you're interested in, and then test it in Earth-based gravitropic and thigmotropic or phototropic assays, and see how does that mutation actually affect the growth of Arabidopsis under those conditions. And the students they use Fiji, freely available software from the NIH, to analyze that root tip angle to vertical. They learn how to acquire their data, analyze it, and present it in figures like this. Always having a control in blue and then their mutant seeds in orange in this case. So they get great experience in data analysis and presentation of findings as well. So I'm going to share with you next a little bit about the actual TOAST database and how that's used. Some of this is data that Melanie obtained from working with me last spring semester in the space biology course, and also this past summer through our summer research program. So she basically asked, which Arabidopsis genes are going to play a key role in the plant's development and function during spaceflight? A big question, right? A lot of researchers in space biology are working to try to identify these genes. So Melanie sort of tackled this with a bioinformatics approach, trying to narrow down from thousands of genes, ones that she was particularly interested in studying this upcoming semester through independent research in the fall semester. So how did she do this? Well, she used the TOAST database, as I previously mentioned, to analyze this GeneLab data from former spaceflight experiments involving plants. Now, these are experiments with all different types of conditions, some grown in light, some in dark up there on the space station, some on the space shuttle, many different experimental variables to look at. She then used a program called ePlant, which provides more additional information on individual genes she identified in TOAST, such as where they're located in the plant. She also looked at programs called String and Netminer, which highlight the associations or relationships between genes of interest. You can start to look maybe at signaling pathways and look at many other key proteins or genes that might be involved. And then she used a database called Signal to look at the mutant sequences of genes and help her identify which seeds she might need to order from the Arabidopsis Resource Center that have the correct mutations in the genes she's interested in. So this slide's probably one of the most important in my talk because it sort of highlights how TOAST works. This is a screenshot of the TOAST database, and when you go through it, there's a lot of information here, but you can start to narrow down what you're interested in. So essentially, she pulled up this one experiment, GLDS 37, a comparison of 4 ecotypes of Arabidopsis in a Brick 19 canister. And she was particularly looking at the wild type Columbia ecotype versus WS2. So you can see here there's a plot that will appear that looks at significant fold changes in the wild type seeds in flight versus ground. She can set filters to narrow down those changes that are significant in flight versus ground. She can narrow it down to changes that happen in genes that would be found just in the nucleus. She could also change it to different subcellular locations, such as genes may be found in the mitochondria or other organelles. So you really, as the researcher, get to choose what you're interested in here in these filters. As you're adding on new filters, there's a toolbar that will appear at the top that keeps track of them. So if you don't like how something narrowed it down perhaps too far, you can always X that out and get rid of that particular filter. You can see here that as she narrows it down, the gene count starts to decrease. So it could start with hundreds to thousands of genes, and eventually through these filters you get to a set of genes that's more manageable. And then the list, the number, the name of the genes are shown down here in the bottom left, and you can actually download that and start to look at them in other databases. A nice thing about TOAST is that you can keep these parameters set and also apply them to other experiments. So we applied them to this GLDS 37 experiment. We could go in and select a different one that's uploaded to GeneLab and start to see if, using these same parameters, do we get the same genes that pop up in other plant experiments that were conducted on the space shuttle or the ISS. So I'm just briefly going to go through a couple of the candidate genes she identified. We don't really have time to get into the details, but you can see some of them here. One is called NERT 1.5. It's a nitrogen transporter in the roots. You can see in these e-plant pictures from that database in red, it highlights where this gene is expressed. So you can see it's very involved in the roots. And it seems to be downregulated in spaceflight compared to ground. You can also see that it's impacted or highly expressed in a lot of conditions of stress. And then she's found that it's downregulated in all these different experiments that were part of NASA's GeneLab. So we showed you that 37A one, but she also found it in 120, 217, and 17. And then she was able to use some of the other plots in the TOAST database to look at how this NERT 1.5 expression changes in the dark versus the light. So that's something else that she's interested in studying in the future. Two other genes that she's working on right now are called MPK3 and PUB24. These seem to be genes that are involved with the plant's response to pathogens and have some role in plant defense. They are both expressed in the leaves as well as the roots of the plant. MPK3, maybe a little bit more in the roots compared to PUB24 here. They're both expressed in response to different stressors that plants might face, abiotic stressors. They were both found in several GeneLab experiments that involved plants. There was some difference in the expression of these two genes in light versus dark. So she found that MPK3 was more significantly expressed in the dark versus PUB24 was more significantly expressed in the light. Then she used programs such as, this is String, I believe, to look at the relationship of these genes. So you could see PUB24 and MPK3 are directly related, and it also allowed her to find a couple other genes that might be playing a role in this MPK3 response that we observe in spaceflight. And she can see that they are co-expressed together in Arabidopsis in this diagram, but none of these other genes are found in other organisms. Using NetMiner, you get complicated diagrams like this, but you can sort of highlight that MPK3 does have a branch here that connects with PUB24. There are a lot of other genes that might play a role in the relationship between these two and why they might be affected in spaceflight. So with that, I'd like to focus on our future directions. Melanie is going to be doing research this semester to do gravitropic and phototropic assays on these mutant seeds that she's ordered from the Resource Center. I want to let you know that this does work in a college level class. We've done assessment to see how effective is this type of lab curriculum at helping students understand bioinformatics data. And you can see here that in gray, I assess the students at the start of the semester, in blue at the end of the semester after they've gone through this laboratory experience. You can see they've gained a lot of confidence in being able to design 3D printed payloads, to use bioinformatics data sets to find their own genes that they're interested in studying, so using that TOAST database. Large increase in confidence in working with Arabidopsis. This is the first and only class at Carthage College where they get to work with this model plant, or any plants for that matter. Increase in confidence in using image analysis software, preparing, and then not significant, but an increase in preparing professional figures and executing the scientific method. Perhaps not significant because they do get those skills in other courses prior to arriving to mine. So with that, I would like to thank the people who've helped fund this research and this course, particularly the Space Grant Consortium in the state of Wisconsin. They've given me a number of grants, such as the Higher Education Incentives Award, to help support this course and the guest lecturers that I invite to it. And then I'd also like to thank Dr. Simon Gilroy and Richard Barker from UW-Madison, who've collaborated with me extensively on the space biology plant research for this lab curriculum, and have let me use the TOAST database and help train my students on the use of that. Lastly, thanks to all of the students who've been involved with this course in spring 28 and 2020. If you have any questions, you can reach out to me. My contact information is here, and I also have all of the resources from the course posted online at this website. Thank you, everyone. Please feel free to reach out at my email, ahenle@carthage.edu, if you have any questions or ideas.